Hi, thanks for inviting me to present you SwissLipids, a knowledge resource for lipids and their biology. My name is Lucila Aimo, and I work in the SwissProt group of the SIB. The SIB is the Swiss Institute of Bioinformatics. It has 67 groups all across Switzerland. The SwissProt group is based in Geneva. It develops knowledge resources for the life sciences and has 24 expert bio-creators, of which just a couple of us work in the SwissLipids project. So today's talk is about how we build SwissLipids. I'm going to show you the website and how we plan to improve it. So how we build it. Well, it all started when Gisou van der Gut and Howard Riesman from lipidomics labs in Switzerland contacted us because they needed help linking their lipidomic data to biology. As you know, when you run samples in your mass spec, you get a list of analytes that cannot always be linked to specific structures and hence the lipid biology. So our approach was to turn the problem around and use biocuration. We capture knowledge from literature of proven lipid structures and pathways in machine-readable forms, and we use it to infer all possible lipid structures and analytes. So SwissLipids can be found at SwissLipids.org. It's freely available, and it's a knowledge resource designed to facilitate integration of lipidomic data and lipid biology. So first of all, to build SwissLipids, we curate the knowledge of lipid metabolism. For this, we use mainly 3 established resources like PubMed for all the peer-reviewed articles, Uniprot as the reference resource for protein annotation, and RIA as the reference resource for biochemical reactions. Both Uniprot and RIA are developed in our group, and they are generalist resources. So in order to make it specific for lipids, we created SwissLipids. Uniprot is freely available and found at uniprot.org. It has over 500,000 proteins annotated, and you can find from the sequences to the function, the variants, all types of information. The RIA reaction knowledge base is also free, and it's available at ria-db.org. It is an expert curated resource of biochemical reactions, not only transformation reactions, but also transport. And all reactions are balanced by mass and charge. Currently, there are about 12,500 reactions, of which about 25% were curated to build SwissLipids. RIA uses the KEBI ontology to describe the reaction participants. KEBI is developed at the EBI, and it's like an encyclopedia of small molecules of biological interest. And it has all the chem information. Mainly, it deals with known chemicals and does not try to cover all possible structures that may exist, but it provides an ontology that can deal with variable knowledge on fatty acid composition, on stereochemistry, and connectivity. So it's very useful to specify the relationship between chemicals and their classes. So to build SwissLipids, we first curate the lipid biology. This means we annotate all the information that we find in peer-reviewed literature on known lipids, including their structures, metabolism, interactions, localization. And from this information, we also get the building blocks that we need, like the lipid classes and fatty tails that we later use for the library of all possible lipids. And these are all mapped to KEBI. So secondly, we build a library of all possible structures, known and theoretical possible lipids. As you know, the building blocks are mainly lipid classes, which we call lipid heads, because most lipid classes have steady backbones with fatty acids attached at different positions. And we create all of these in KEBI. And the other building blocks are the fatty tails attached to the lipid heads. And these we also get from the literature. They are mainly fatty acids and fatty alcohols. So then in SwissLipids, we enumerate the lipid structures by combining the fatty tails with the lipid heads. following a set of rules, and we generate all possible structures resulting in an in silico lipidome. And here you see a selection of some of the 500,000 structures that we have in SwissLipids and that are linked to KEBI, RIA, and Uniprot. One of the most recent curation efforts we did was to digitize SphingomApp. SphingomApp is a knowledge resource for complex glycosphingolipids that was developed by Alfred Merrill Jr. and collaborators. And thanks to the curation, now we have enumerated over 400 classes of complex glycosphingolipid classes, and I will show you in the website one example. So last, to build SwissLipids, we create an analytical hierarchy. This is, we work up from structures to build a hierarchy of the possible lipid analytes that you might get from the analytical method used. For this, we use the shorthand lipid notation that is compatible with the recommendations of the Lipidomic Standards Initiative. We will also see in the website how we use this hierarchy. So in summary, to build SwissLipids, we curate the lipid biology about known lipids from the peer-reviewed literature. We get the building blocks that we need to generate the in silico lipidome and have all possible lipid structures, known and unknown, which thanks to the ontology can then be linked back to the biological knowledge. So let me show you the website. If you go to SwissLipids.org, you will land in this homepage that has a search box. You can enter your term in this search box, like a lipid name or a protein, or you can use the advanced search and enter a smile string or an inchy key if it has one, or the formula or the masses. You can also browse the website. It can be by lipid classes or by lipid analytes. In this example, we browse by lipid class, so we get a tree of all the different classes, and as we go selecting, we reveal more class members. So as I told you, we now have the complex glycosphingolipids. So if we choose sphingolipids, we see the gangliosides, and if we click on ganglioside, we can see all the different possibilities with the different sphingoid bases as well. And if we click on this one, then we will see all its class members. Next to each structure, there is a set of colored icons. Here you can see it bigger. When these colors are highlighted, it means it has a manual annotation. In this case, it has cheminfo, classification, and metabolism. So if we select this particular lipid, we will see its 2D structure with both the glycan moiety and the lipid moiety. It has a list of synonyms and cross-references to other resources. And if you look into metabolism, you will see the reaction where it participates with the RIA ID. You can click the RIA ID and go to the RIA website to see the reaction. And you can also click on the enzyme that catalyzes this reaction and see which other reactions are catalyzed by this enzyme and have been annotated in SwissLipids. All of our annotations also have an evidence tag that you can click to see the reference and a piece of text to support such an annotation. The other browsing mode is by lipid analyte. When you run the sample, you can get a list of analytes that will only give you the class and the sum composition. So you can enter the class and the number of total carbons and total double bonds and search, and it will search through the hierarchy and provide a list of the matching structures. Again, these structures have a set of colored icons that when highlighted indicate that there is manual annotation. So if you click this one, you can see this lipid participates in this reaction. And again, you can go to RIA to see the reaction or see the enzyme and the evidence tag. The last tool I want to show you of the website is the ID mapping tool. So if you already are using other resources like lipid maps or KEBI, you can enter those IDs in this box and choose to map it to, for example, if you want to see the enzymes, you choose Uniprot KB and you map it. And then you get the list for that lipid, those lipids, and the enzymes that have been annotated in SwissLipids for such lipid. Okay, and last point is how we plan to use Uniprot KB to build an improved version of SwissLipids. So Uniprot is the reference resource for protein annotation. It describes the function and it also describes the catalytic activity. While previously this was only a text line or an easy number, since end of last year, it started using RIA. And here is the last publication that I invite you to read. So now all the catalytic activity is described using RIA, like we do in SwissLipids. That means that now you can search Uniprot for your lipids. So you can enter here the name of the lipid or the KB ID, or if you have the INCHI key, you can also enter it here and search for your enzymes. So we plan to build the next iteration of SwissLipids using Uniprot as our annotation source, because there are many more biocurators working in Uniprot, and we will cover more proteins, more papers, and species. And so we plan to leverage the knowledge of Uniprot. So in summary, I hope I have convinced you that expert biocuration is fundamental to integrate lipidomics with biological knowledge. SwissLipids is freely accessible and available for anyone to use. It provides a range of search and browse options, as well as an ID mapping tool to other lipid resources. It comprises a library of over 500,000 structures from more than 600 lipid classes, which are linked to biological knowledge. We plan to continue growing the in silico lipidome and use Uniprot to design more lipid structures in SwissLipids. So if you want to see your proteins, reactions, or lipids curated, please contact us. You can do at SwissLipids@sib.swiss. This is the SwissLipids team. We have some curators, the software development led by Robin Lieschi, and our PI, Alan Bridge. And also I would like to thank the support of the RIA team and the funding agencies. Thank you for your attention.